Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is January 30th, 2026, and we are back from spring break and continuing our series of conversations with neuroscientists. Today, I'm happy to have one of our own here, Fidel Santamaria. Fidel is professor of neuroscience, developmental, and regenerative biology here at UTSA, and he's not new to the podcast. In fact, he was our very second ever podcast guest back in October 2007. And at that time, he was a new assistant professor. And I recommend that podcast. It has better sound quality than a lot of our early attempts, which vary in sound quality. And it is still timely. The topic that was discussed, which included what do we need to make a working model of a part of the brain? What pieces of it do we have? What pieces can we leave out? What pieces are we struggling to get? And how much it hurts us to not have some of them. And he was doing it in the context of the cerebellar cortex and trying to define a sort of point spread function for activity in the cerebellar cortex. I hope that's a fair statement. And so since then, Fidel's been a frequent participant in the podcast, but we have not caught up with him on his own scientific journey over the past 18 years. And this is our chance. So welcome, Fidel.
Thank you, Joe. Thank you for having me again.
I'm Charlie Wilson. So, Fidel, at the end of our previous podcast, we got a little preview of where you are going because you talked to us about sort of unexpected aspects of diffusion, empirical features of diffusion in the intracellular compartment and in the cell membrane that didn't follow the usual kind of expectation that people have for simple diffusion. And we also talked about why that happened, at least as the understanding was at that time, and a little bit about what the implications are for synaptic plasticity, because synaptic plasticity depends on diffusion of molecules around in the cytoplasm. And you have worked a lot more on that. And it would be a good thing to talk about. But I kind of like to ask you instead about a newer project you've had, which is on the kinetics of voltage-sensitive ion channels and their temperature sensitivity. So I'd like to try to start by explaining the kinetics of ion channels in the school way, in the way you would explain it to an undergraduate, which is that's the level at which most of us know it, because that's how most of us learned it.
Right.
So after I go through this, you can tell me what's wrong with that and where we should go with that and what's happened, or actually what lies underneath all of that stuff. So here it is. Here I go. So the idea is that ion channels open and close in response to voltage changes, and that the opening and closing is determined by two rate constants, a rate constant of opening and a rate constant of closing. And both of them are voltage sensitive, and they have an exponential dependence on voltage. And they differ in the sign of whether they're opening or closing under the voltage at any particular voltage, and the location of their point of shift. So they are offset from each other so that opening may win at one voltage and closing may win at another. And so that's what makes them voltage dependent, but they're also exponentially dependent on temperature. But opening and closing in our normal formulation, opening and closing rates are both equally dependent on temperature and in the same direction. And so what happens is that both opening and closing are slowed down at low temperatures and sped up at high temperatures. So for any one ion channel, temperature kind of determines the time frame. It's just saying, is time going fast or is time going slow? And if all voltage-dependent channels had the same temperature dependence, then the whole neuron would just speed up or slow down. Action potentials will get broader or narrower and go faster or slower as the temperature changed. But it's not like that exactly because different ion channels have different temperature sensitivities. And so that's the simple view of it. Temperature is basically determining whether an ion channel is working in slow motion or fast motion.
Mm-hmm.
And of course, in some animals like squid that live in the sea and don't regulate their body temperature, then they are sometimes in slow motion and sometimes in fast motion. And that we have this idea about cold-blooded animals like frogs,
Mm-hmm.
that become slow in the cold. So to start, could you start by telling me from the fundamental physics of molecular function why an ion channel would be exponentially dependent on temperature and why it'd be the same for opening and closing? And maybe that's not even true, but that's what we think. Why do we think that?
Well, what we think is like any opening and closing, for any voltage, you will have an energy barrier. Still, there's imagine the voltage channels are not in the vacuum. They're being bombarded by temperature, that is the collisions of water molecules. That's the main carrier of energy in all living organisms. And then when you have that, it's affecting a little bit how the channels are going to respond to voltage changes. And the idea goes that as the temperature increases, well, the more water, what that means is that water molecules have they move faster. There are more collisions per second or per nanosecond, and that just transfers more kinetic energy to the molecule. Things vibrate more, and it's easier to open. Okay, and that distribution, if you look at the distribution of energies, that kind of can follow like an exponential.
Easier in a probabilistic way.
Mm-hmm.
I mean, the energy of activation, is it actually changing at temperature? The energy barrier?
That is the idea. As you increase temperature, the energy barrier, it's easier for anything, just as a frog. When it's cold, they don't jump as fast. When it's warm, they will jump faster when they see you approaching. That is the idea, actually, in explaining the speed of reaction of some animals. That has been the issue is that has been generalized to molecules, right? And then a molecule that responds at a certain rate, at room temperature or body temperature, you just have to increase the temperature so the reaction happens faster. So whenever you change the voltage, the activation of the channel happens faster and faster and faster. And it only depends on the average temperature, the average collision of molecules towards your protein, and that's just basically an entropy. That's entropy. It only depends on the entropy of the system, how fast things are moving, how disorganized things are, or well mixed. And that's kind of our current idea. So if you just go to theoretical aspects, it's like, well, I have something that it has a reaction rate of 5 milliseconds. Right? I mean, I try to activate it, it takes 5 milliseconds to react. Well, if I increase the temperature, I increase the temperature by 10 degrees, it's going to be a factor of 2 or 3 faster. And then if I increase it by another 10, it's going to be like that. And unfortunately, the theory says if you continue, you can do that infinitely. That's kind of how we've been doing it at the level of undergraduate biology or undergraduate physics, and it's called Arrhenius function, and it has to do something with some coefficients called Q10. And everybody in the biosciences, including neuroscience, use that approximation, and that's that the idea is only depends on temperature and not on the properties of the molecule do not change, and that's the assumption there. Is that?
Yeah. So I get it because it's kind of a rule of thumb that enthalpic things don't care much about temperature as much as entropic things do. And I'm used to thinking about the main entropy component is hydrophobicity. And so things that are held together by that become weak when it's cold and become strong when it's warm. Whereas things held together by covalent bonds don't care very much about that. So I'm just kind of trying to figure out what's the relationship between energy of activation and entropic interactions in the molecule. Is that a thing?
Well, I mean, instead of thinking about voltage activated channels, I like to think about, if you want to generalize it and any enzymatic reaction, I think about a game of pool. In a game of pool, you have your balls, and then imagine that you can put more balls and you can make them move faster. Entropy is just going to make them collide on any point on the surface faster over a period of time, or more frequently over a period of time. That is a probability of reaction. If that, you instead of thinking about the balls, you think about the parts of the molecules inside the protein, it's basically the same. That's probably an actual formal analogy.
Good, that's great. Because the parts of the molecule inside the protein I'm thinking about are the voltage sensors. These are charged parts of the protein that move under the influence of voltage.
Right.
And so as they heat up, they are starting to move on their own more? Is that what it means?
That could be, that is a reasonable interpretation. Now, when I went to college, and probably a lot of our listeners, when they went to college, there was this idea that proteins were kind of semi-monoliths. They were just like these mega Lego blocks that will just reposition depending on some almost mechanistic in like classical mechanics. You apply this voltage or this pH and the thing opens or it closes, right? Luckily now our students, they can see full atomic resolution simulations, and you can see that everything is shaking. You apply a little bit more temperature and the energy that is being delivered to the plasma membrane or to the cytosolic or extracellular part of the proteins affects the shaking of the entire molecule. So it is not monolithic, and it will be affecting the energy barrier of not only the reaction, but any other reaction in which the protein, the enzyme is involved.
Okay, so great. So I follow that. I hope everybody follows that. And that explains why there would be a temperature dependence of the rate of both rates, because both opening and closing would be affected by that. But tell us what's wrong then with our regular formulation, because it just predicts this nice exponential relationship between the time constant, because both opening and closing are equally changed, which means the time constant of the whole thing, of both opening and closing, are shifting together. So we should just be able to see this nice, simple relationship between temperature and time constant. And that's built into almost every neuron model that anybody has made since the Hodgkin-Huxley days, where they built it in for their squid model in order to extrapolate what would happen to their channel at other temperatures. And so does that work?
Well, I mean, let me go back. Let me try to see if this works. Let me go back to the monolithic idea of proteins, or semi-monolithic idea of proteins, that it was prevalent until the 1990s or the beginning of the 2000s, in which it was basically like a Pac-Man. It's either open or close, and then the energy is just going to allow you to open faster or more frequently. It doesn't matter, right? And then the idea of what I told you, that now if we try to model every single atom, we can see that the energy is being transferred all over the structure. It's not like a monolithic skyscraper. is more like one of those toys that kids play with that is that jumps around and is like don't remember the name of it. My daughter had one, but it's always shaking, right? Or one of those guys, the little thingies that sell cars that
Transformer.
well, like a transformer. Anyway, it's a problem coming with metaphors. The point here is that every part of the molecule is moving. So now, if you're thinking, oh well, does it matter only when we change the temperature from physiological values to boiling? Or is this shaking affecting the properties of the molecule of the enzyme within physiological ranges? And that is an important question because it has been obviated that it doesn't, right? But if you just ask, does it matter? What would it change if we believe that the molecule is moving? We change 2 Celsius degrees. Is it shaking the same way? Are there new hydrogen bonds being disassembled or assembled depending on the temperature? We know that happens with water, so why not with the enzyme? And if you say that, then if you say that within physiological ranges, then the enthalpy and the free energy of all reaction rates, not only the activation, but in particular because we care about voltage activated channels, could be changing. Okay, so that is just accepting a potential hypothesis, and you can ask, is that real? Have people observed that?
So that would mean that the temperature sensitivity would be varying at different temperatures. So
yes.
It wouldn't be lying on the same exponential curve at one temperature and another.
That's true, because the idea of the exponential, like a fixed exponential dependency, or the exponent of the power of the e to the x value, that x value is assumed to be constant and only affected by temperature, the entropy. If you believe or you accept the hypothesis that maybe within physiological ranges that the structure of the protein is changing, then the value of the activation energy could change. And that's just from looking at what our present understanding of proteins in the natural range of values in temperature or pH or anything like that. And then you can say that, okay, well, if you go outside neuroscience and ask in the a general question, have people seen a temperature dependence of the activation energy? And a lot of people have seen that. And evidence has been accumulating over the last 30 years. Is not at all that this is the idea that this is fixed, that it's only a single time constant, is not applicable to enzymes, to large proteins. It might be true for ion channels, for water, for ions, for water, but for large proteins, it might not. We cannot assume that these things are monoliths. They're just like, they're just.
So about how, it seems like an easy thing to test. You could just take some ion channels and measure their kinetics at different temperatures and look for the deviation.
Yeah, and if
and
you go
what do you get?
So to the historical record, if you just do a meta-analysis of a lot of very detailed, and I want to be careful about here, and I don't want to over-criticize, but people went and did these experiments, and you could see in these experiments some curves that didn't fit a line when you do that in a semi-log plot. But people start saying, well, this is when the channels or the membrane started to denaturate, and then they see a curve, and then they stop the experiment.
So this happened at warm temperatures, and so it was easier for them to say, oh, that proteins being killed by the heat.
Yeah, but it was happening at 40 Celsius.
Which is not that hot.
And even now we know that even at 70 Celsius, proteins do not really denaturate at that. They can come back from that bad situation and keep working normally. So, because that was the second assumption. Whenever this idea that the single exponential activation breaks down, is because the protein is breaking apart. And again, that's not necessarily true. And there has been a plethora of evidence across the biosciences that what we have to assume is that even at a few degrees changes, the conformational structure of the protein in the ion channel is changing, and therefore the energy barrier is being modified. And if you go back to fundamental thermodynamics, well, you have your pressure, your volume, your temperature. But now that we're in the cold months, people try to boil water, and you can put a lot of heat into water, and it doesn't change volume for a long time. That's the heat capacity. Water has and that's also from Bio 1. Everybody at the Bio 1. Everybody learns that heat capacity. Well, proteins, enzymes, and ion channels have heat capacity. So the energy that is being, you change the energy, either the heat up or down. Maybe the volume of the protein is not changing, but the heat, all that is being absorbed in the heat capacity, in the bonds, right? It could be anything, van der Waals, electron pairs, all that stuff, okay? But that is affecting that could be affecting the energy barriers. And if we just write down with entropy and enthalpy and how that can change, we can come up with formulas, or some other people that we have used their theory, we can come up with formulas that then replicate all these nuanced curves that people have seen that deviate from the original assumptions from Arrhenius that have applied to sodium, calcium, and potassium voltage activated channels.
This is good, because I think when we, as an experimentalist, when I have a theoretical curve and I have a bunch of points that follow the curve for a while and then they deviate from the curve, I usually come up with some kind of explanation of why I did something wrong for these points, and they're off by a little bit, but really the curve is the real truth, and that my data are flawed. And actually that argument sells really well.
Yeah.
Because people really want to believe this theoretical curve, and they doubt your data.
Yeah.
Because it's just your data.
Mm-hmm.
So it's a normal thing for us.
It is normal. Yeah. It's just, and people had not observed. It was good enough. It's just that now we can do experiments, and we can understand what's happening. I mean, a lot of these experiments were done when the idea of proteins and the structure of proteins were just being elucidated.
So if you have an alternate curve that is theoretically grounded, and your data fit that one better, it's way more inviting than to just have one curve in your data fall off a little bit. So there isn't a new curve that we could use.
Yeah, so we proposed we incorporated this. This was a theory developed in another field in the biosciences, macromolecular rate theory. We brought it into neuroscience, and we demonstrated that, look, I mean, we can parameterize the theory with experimental data from a wider range of experiments, and then we can go and start explaining things that were not explained in a unified way, like how is that the reaction rates or the sensitivity to temperature measured by the Q10 coefficient at different temperatures is very high in animals that live almost at 0 Celsius. And that is consistent across all organisms. And that is true also for enzymes, like anywhere outside neuroscience. That's why we realized by going outside neuroscience to try to find an explanation.
Oh, well, neuroscientists have studied animals
Yeah.
that live at almost 0 degrees.
Yeah, like fish
the
in the Arctic.
giant axon of the squid was all done at 6 degrees centigrade.
Right. And if you go with different species of squid, they found different temperature dependencies, thinking, we haven't tested all the predictions from our model there, but we were able to replicate the different temperature sensitivities of these different species that live at different depths in the ocean that are related to different temperatures. And we can put them all within the same theory. So that kind of encouraged us to think maybe we're not doing something wrong. What is happening is like all these proteins have kind of, and some people outside the biosciences have been saying, there's like a universal dependency on temperature of all enzymatic reactions, that at a certain value, they start decaying. That is their optimal temperature, but that is not related necessarily to the denaturation. It's not that the proteins are unfolding, it's that there's some balance between entropy, enthalpy, and free energy that then some of entropy will win or enthalpy will win, and then that allows reaction rates to increase at a certain ranges and then decrease. And we have proposed that decrease is ecological, that's a way for reaction rates to protect themselves from overreacting.
Otherwise, my nervous system sped up to the point that it was crazy.
Calcium poisoning, the cell will just let all the calcium flow in. And, if you go to ecology, people have asked this question. Well, most of the organisms in the animal kingdom, or most organisms, they do not control their temperature. And my example is like a fly or a mosquito, depending where you live, will bug you from 70 Celsius to 110 Celsius.
If you live here, you got both.
Both, exactly. And it will be equally difficult to kill them. Okay, and researchers have asked, well, what is there some kind of mechanism? It could be a complicated metabolic mechanism that compensates for the changes in temperature. But here we're saying, well, it could be the same. The proteins evolved to exploit these properties to self-regulate with temperature. And I think it has also consequences for other aspects of neuronal function. But taking into account these just changes, minute changes in enzymatic shape or not shape, energy, can have tremendous effects on the activity of the entire neuron. And when you put that in the context of a network, you could even have much more larger effects.
Yeah, I'm thinking about conduction velocity of axons, which is very much dependent on the kinetics of the sodium channel and the potassium channels. So I'm imagining that if my arm gets warm, that the action potentials start traveling down my arm faster. And if it gets cold, they go slower. And sometimes I kind of feel like that, like my reaction time gets long when it's super cold. Is that a thing? Is that really this?
I don't know about the specific example, because that could be like muscles also, like
muscle.
Or it could be diffusion.
Muscles are also determined by the same activation.
Yeah, but with respect to that, I haven't spent too much time thinking about that particular problem, but I've been thinking about, for example, fever. The connection between high temperature and seizures hasn't been there's no clear. There are some papers that say it is clear the mechanism, and some papers that say there's no clear mechanism. Could the seizure arise from the slowing down of some molecules because they're being heating up and now they start slowing down, and then that allows synchronization of the network? It would be a totally different way of understanding how a seizure related to high temperature and heat stroke, for example, arises. In terms of reaction times, that would be interesting to actually look at this. When we did our analysis, we had a bunch of papers that were reporting activation, but of the bundle of action potentials. But because we were interested only in single channels or the closest thing to single channels, we didn't use them. But we have the data somewhere.
It doesn't happen too much in the periphery, but in the brain, we think timing of arrival of signals from different places in the brain is important.
Right.
So some signals get there first and get priority. Sometimes it's a feed-forward inhibition is sometimes faster than excitation and prevents over-excitation from happening, or at least that's one of the things we hypothesize is happening.
Mm-hmm.
And so if these kind of timing of signals from different brain areas converging in some other brain area is important, then I would worry temperature would mess that up a lot.
Yeah, but there's evidence that the temperature of the brain can be regulated in different areas of the brain, and there's interesting data that it could be affecting how the excitability of large chunks of the brain. But nevertheless, yes, you could have fast processing in some areas and slow down in some other areas by just changing the temperature by 2 degrees. I'm not saying that is an entire, it could be kind of a neuromodulator temperature to start thinking of temperature as a neuromodulator of network activity. And that could be possible. On top of that, we already know that there are some channels that are temperature sensitive, TRP channels.
Oh, yeah.
But excluding that, the temperature could still be a neuromodulator in processes that, as you were proposing, that depend on millisecond timing, and I think that's something that we would like to explore.
But it sounds like the relationship you're discovering sort of flattens out the temperature dependence at higher temperatures and keeps it from continuing to climb, which would seem like maybe it would protect the timing mechanisms from high temperature a little bit, because everything is starting to flatten out. Those curves are all starting to flatten out at high temperature.
Mm-hmm.
Is that?
Well, they start going down. So it's not just flattening out. They start going down.
Well, or before they do that, they quit climbing as such.
Yeah, exactly.
So they start going down at temperatures that are quite high.
Mm-hmm.
Maybe temperatures that don't happen much.
Well, what we found interesting, and we haven't explored that, is like if you take the derivative of the functions that the functions are plotted as reaction versus temperature, and if you take the derivative of that, the highest derivative is around in the physiological range. So my point there will be like it is the change.
So the changes will be biggest.
Yeah, exactly.
Kind of near the set point of temperature.
So that's enticing. That's something that the problem there is, how do we measure these small changes? I mean, I like to do theory, but I like to go hand in hand with experiments, and I don't like to get too much into fantasy land, right? So, and through other collaborations, and other people that I've talked to, is like trying to measure temperature at the pixel level when you're trying to image a piece of the brain is not a trivial thing. Okay, there are some cameras that can be parameterized to report that, but but maybe it's going to be in a slice, and it has to be a very, very particular kind of setup to induce heat changes in one part of the tissue and not the others, and then try to test this hypothesis. But we will see. I mean, we will see.
Okay, thanks a lot, Fidel. We didn't get to talk about anomalous diffusion.
Oh, well, next time, 18 years from now.
We'll do that another time.
All right, thank you, Charlie.
Thank you. This has been Neuroscientist Talk Shop.